In this video, we're going to go into more detail about sexual reproduction in true ferns. And this is referring to the specifics associated with the alternation of generations. As mentioned previously, the dominant generation in the alternation of generations for true ferns is the multicellular diploid sporophyte stage. So what you're seeing here in this picture is the sporophyte of the true ferns. And ferns, these true ferns, have these sporophylls. And remember that a sporophyll is just a modified leaf that contains sporangia. So these little kind of clusters on the underside of the palm frond, these are the sporangia, and they're actually contained in these things called sori. Sorus would be singular. And in the sporangia in these sori, meiosis occurs. And remember that meiosis, we are changing the ploidy of the cells. So a diploid cell is going to go through meiosis to produce haploid daughter cells. And in this case, the sporangia, the cells in the sporangia, are going through meiosis to produce unicellular haploid spores. If the unicellular haploid spores find a suitable place to germinate, they will go through mitosis. Remember that the ploidy and mitosis of the cells stays the same. So if a haploid spore is going through mitosis, it is producing daughter cells that are also haploid. So during germination and mitosis, a multicellular haploid gametophyte develops, and this is known as a prothallus. And the prothallus has this kind of heart shape to it. You can kind of see it here in this picture. And on this one, it's a little bit harder to see it on the male gametophyte picture though. This prothallus develops into male and female gametangia. In males, remember the gametangia is known as antheridia, or singular is antheridium. And in females, the gametangia are known as archegonia, or archegonium is singular. And then the specific archegonia structures are these little spots kind of in the middle of the prothallus here, and the antheridia are these spots here. And then cells within these male and female gametangia are going to go through mitosis. Once again, we're starting out with haploid cells that are producing haploid cells. And they are going to produce these unicellular, so single-celled haploid gametes. And so the antheridia is going to produce the male gametes, and the archegonia are going to produce the female gametes. And the male gametes are these sperm that have a flagellum, and they can swim to the egg, which is the female gamete. And so it swims over to fertilize the egg. During fertilization, the gametes fuse together. So remember we talked about the male gamete being a sperm, and the female gamete is an egg. And the sperm swims over to the egg and they fuse together. Remember, the sperm is carrying one set of chromosomes, so it's haploid, meaning it has one set of chromosomes. And the egg is also haploid, so it has a set of chromosomes. And so when they fuse together, it creates a zygote, which is a single cell, but it is diploid because now the chromosomes from both the sperm and the egg are in the nucleus of the zygote. Now this zygote is actually retained in the archegonium. And so the archegonium, remember this is the female reproductive structure. And the archegonium is essentially going to act as an incubator for this zygote as it goes through mitosis in order to develop into an embryo. On the previous slide, we left off talking about this unicellular zygote that is diploid because it has chromosomes that it got from the egg and the sperm that are now paired up in it. And remember that the zygote is retained within that female reproductive structure, the archegonium, and this helps to provide protection and nourishment. The zygote is then going to go through embryonic development, so it's going to go through mitosis to turn into a two-celled structure, and then it's going to go through mitosis again to form a four-celled structure, and then that's going to form an eight-celled structure, which forms a 16-cell structure, which forms a 32-cell structure, etc., 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 until a new sporophyte is produced. And then the cycle restarts, because remember, we started with the multicellular diploid sporophyte when we started talking about this alternation of generations. So let's do a quick overview of the alternation of generations in true ferns. So we'll start with the sporophyte, which remember is the multicellular diploid structure. And the leaves of the sporophyte are known as sporophylls, which have the sporangia on the underside of their fronds. Within the sporangia, meiosis occurs, because remember that the sporangia, the cells in the sporangia are diploid, but the spores are haploid. Those spores can then go through germination, where they go through mitosis and they create more haploid cells to form the gametophyte. And the gametophyte has this kind of heart shape to it. And within the gametophyte, there are male and female structures. There's the antheridium, which is the male structure, and the archegonium, which is the female structure. The archegonia produce eggs, and the antheridia produce sperm, which are the gametes, and they produce these gametes through mitosis, starting out with a haploid cell producing additional haploid gametes. Fertilization then occurs where a haploid egg fuses with a haploid sperm, producing a zygote, which is now diploid, but still unicellular. Remember that the zygote is contained within the archegonium, for protection and nourishment. The zygote is then going to go through mitosis during embryonic development, producing more diploid cells to then produce a new sporophyte, and then the cycle continues.